Good afternoon. It's a great pleasure to be able to talk about this. Translational medicine is something I think we all feel passionately about, otherwise we wouldn't be here. Particularly because whilst I love the science, it's all directed to try and improve the lives of patients. Oops. Right, so what I'm going to talk about is some work that has been going on for many years. I wish I could give the denouement just to start with. I can't because the work is unpublished. Until it's been published, it would be wrong to present it. I'm beginning to wish we'd sent it to Science Translational Medicine. It might have been published by now, but never mind. And I've taken a bit of liberty with the title. It was From Concept to Clinic. I'm taking it from conception to clinic now. So I've been fascinated since really being a junior doctor by patients who've got mitochondrial disease. It's a fascinating group of genetic conditions which can affect children of any age, patients of any age, from very young to very old. It's fascinating as well because it's got two different genetic bases. It's the only genetic disease or groups of disease where you've got both the nuclear encoded genes and you've got the mitochondrial genes. And there's about 1,300 nuclear genes which encode mitochondrial proteins. There's been genetic defects in about 300 of those. So it is complex to work out, but there's also the mitochondrial DNA. For the rest of my talk, we can ignore the nuclear DNA, we can focus on the mitochondrial DNA. Mitochondrial DNA disease does tend to affect more adults than children, but it can affect children, and it has particular genetic characteristics. It's also really interesting because, like other mitochondrial diseases, it can affect many different tissues. So whilst I'm a neurologist, patients on the whole don't die so much of a neurological disease, of either cardiovascular disease or other complications of the mitochondrial disease. So it's fascinating clinically and fascinating genetically. Despite us knowing about mitochondrial DNA disease since 1988, when the first genetic defect was described, I don't think we're any closer to curative treatment. Gene therapy for mitochondrial DNA disease is very difficult because conventional vectors just simply won't get into the mitochondria. So it's a real struggle. But this is where this story starts. So this is a lady that I saw back in the 1990s, and I've told the story to the ECR meeting this morning. This is Sharon with her son Edward, who has a Sharon's affected, and Edward is affected with a severe form of mitochondrial disease. He died maybe 7-8 years after that photo. Sharon has always been very good at sharing her story. She's actually had 7 live pregnancies. 6 of them died within the first 36 hours of birth with severe lactic acidosis, suggesting a mitochondrial defect, and then Edward, who was severely affected. So my clinic was filled with people like Sharon, with family history of mitochondrial disease. And what was so interesting about mitochondrial DNA is that it is only passed down the maternal line. At the time of fertilization, you've got an egg with about 500,000 copies of mitochondrial DNA, and you've got a sperm coming along with about 10, and they tend to get destroyed. So all your mitochondrial DNA comes from your mother. That's been shown in numerous studies, and of course, genetically, it's very interesting when you're looking back to an African Eve, you can chart it through the mitochondrial DNA. So what seemed to us a really important problem was, if you can't cure it, can you prevent it? And how might you be able to prevent it? Well, the actual solution for this was in a paper back in 1983. And this is something that I think, this is why you need clinical academics, because a scientist would never have thought of doing this experiment because it's already been done in mouse embryos, which is transferring nuclei between different embryos. It's only because there was a pressing clinical need did we start to think about this. So what is the principle of this? Well, you have a woman who has mitochondrial DNA disease, which is this oocyte or egg. It's fertilized, and you get 2 pronuclei. You get a donor egg, which is normal mitochondrial DNA. You fertilize it, you remove the pronuclei, you transfer these 2 pronuclei to the enucleated egg, and you've got an egg which has got the nuclear DNA of both parents, but the mitochondrial DNA of the donor woman. So in theory, that should be a way to prevent the transmission of mitochondrial DNA disease. As I pointed out this morning, we had that initial idea back in the 1990s, but it was not allowed. We were not allowed to do it because of the Human Fertilization Embryology Act. And the reason why it wasn't allowed in the Human Embryology Act is because they wanted to prevent cloning. Any sort of nuclear transfer could effectively have been a way of doing cloning. So it was sensible to stop it, but if we ever want to see this as a treatment, we needed to be able to change the law. And this was a long journey, and I'll just highlight the timeline of this. We first started talking about this back in 1998. We had discussions with the Human Fertilization and Embryology Authority, that's the HFEA, to allow research on human embryos to prevent mitochondrial disease. We got our first research grant in 2000 to do some of this manipulation in mice, because of course we weren't McGrath and Salter. We hadn't got the technology to do it. And in 2005, and I was reminded of this last night, was we actually put in a license to the Human Fertilization Embryology Authority to do work on human embryos. It was turned down, and we had to get legal representation to fight that, to get that license. In 2010, we published our first paper showing it was feasible to do in human embryos. At that point, a very, very wise person, I'm all for, oh, we've got to get the science done, and then we'll bother with the legislation. Somebody very sensible, Alison Murdoch from the Fertility Centre in Newcastle, said, you've got to start now thinking about changing the law. So if you're thinking there in 2010, you'll see where our journey went in terms of changing the law. There were bioethics reports, there was scientific reviews, a public consultation, regulations laid before Parliament, and in 2015 there was a vote in the House of Commons and the House of Lords which approved it. And in fact, it came into law at the end of 2015. One of the things is that when you get involved in something like this, something that few of us will ever get involved in, and I'll never ever be involved in again, you don't realise how much help you need. And in fact, last night, this was us and Jamie Oliver's restaurant in Catherine Street, with the people who are all involved in getting this through Parliament. From people like James Lawford Davies, who's the lawyer that dealt with it, for people who work for Wellcome. It was a fantastic group of people. Liz Curtis from the Lilly Foundation, the patient organisation. It was just a fantastic group of people, which, I mean, in effect, changed the law. You know, it was this group of people with support from outside. So in 2016, we applied for a license from the regulator. And then in 2017, the license was granted. So I just want, this is where I think it really is important to just think about this in a way So you've got a license and you've got patients, but how the heck are you going to fund this? I'm going to remind you later that everybody that goes through mitochondrial donation has to go through 3 rounds of IVF to collect eggs. So just imagine the cost of that. They've got to come up to Newcastle to have it done. You've got to pay for the donor. Who's going to pay for this? And patients with mitochondrial disease can't afford tens of thousands of pounds to go through treatment. And I have to, again, I was praising the NHS highly specialised services this morning. I got rung up by the head of the highly specialised services and says, Doug, we can't fund this. We'd love to fund this, It's got through Parliament, but we can't because it's first in man. But if you can get Wellcome to fund a clinical trial, we'll do the costs of running the clinical trial. And Wellcome, bless them, in about 10 minutes said, yes, we'll fund a clinical trial. So what have we been doing since 2017? Well, I'm going to try and explain, and I'm going to try and explain just how challenging this has been. So if you've got a family, a woman with mitochondrial DNA disease, what sort of counselling can you give? What sort of support can you give? So you can explain, voluntary childlessness, adoption, egg donation, prenatal diagnosis, which is applicable to some women, or pre-implantation genetic diagnosis. And it's really important that I explain a little bit about this before I go on to what's happened with mitochondrial donation. So mitochondrial DNA genetics in some ways easy, and in other ways it gets a lot more complicated. So if you think about it, a cell has literally hundreds or thousands of copies of mitochondrial DNA. So if you're homoplasmic wild type, all your copies are normal. Or if you're homoplasmic mutated, like Sharon is, they're all abnormal. But in the middle, you can have a mixture of mutated and wild type. Now, does that matter? Well, it definitely matters, because if you've only got, these are functionally recessive, so if you've got a low number, you're not going to be affected. If you've got a high number of mutated forms, you're going to have disease. It's further complicated because when eggs are formed during embryogenesis, and a significant proportion of this population will have eggs that were formed, there is a genetic bottleneck. In other words, the copies of because there's such rapid expansion of eggs, the copy number of mitochondrial DNA molecules goes right down, and that means that even though you might have 50%, in the woman, you can have some eggs which have got very high levels and some eggs which have got very low levels. And clearly, if you do pre-implantation genetic testing, which is actually doing embryo biopsies, then you can get an interesting result. And this is perfectly highlighted by, this was the first pre-implantation genetic testing in the UK. A lady we looked after, she had 50%. Her child that died had 98%, died in the first year of life with severe mitochondrial disease. The embryo put back had 5%, so the child is effectively normal. So pre-implantation genetic diagnosis does have a role. And then we bring on mitochondrial donation. So where does that fit in? And how can we provide a clinical service that is not directive to women, that is giving them plenty of time to consider the complexities of this and making their own choice, on the knowledge that pre-implantation genetic diagnosis involves an IVF procedure, not something without risk, and mitochondrial donation includes that, and it's also completely untested in humans. So what we've set up is this setup. So we have a mitochondrial reproductive advice clinic. It's run by our nurse specialist, a fantastic clinical clinician. A whole load of things are discussed. You're not in and out in 5 minutes, I can tell you. This is a long discussion. Fitness to fertility is an important one because these women on the whole tend to have mitochondrial disease, complications of pregnancy, and something which is really important is to make sure that you don't broadcast that you're going through it. Then those women that want to progress to the next stage, because not everybody does, it's not necessarily relevant to everybody, will then go to a clinic which is run, was run by Jane Stewart, now by one of her colleagues, and my colleague Bobby McFarlane, where they actually have a much more in-depth conversation about what might be feasible, whether it be mitochondrial donation, PGT, pre-implantation genetic testing, or egg donation. We've got to think about donors. Now, fortunately, there is a way, and not many countries. This is trying to be brought in Australia, the only other country that's legalized this, and they have huge problems because they don't have an HFEA about ethics. So we've got to get donors. Not only do we have to get donors, we've got to prove that they don't have mitochondrial disease. So they've got to have several tissues sequenced. And then what are you going to do about looking after the mothers after they get pregnant? I don't think any of the people that are going through very few people that are going through the program are from the northeast of England. I mean, this is a nationwide offering. There are concerns. These women already have mitochondrial disease or may have. They don't want to be over-medicalized. So actually, a lot of this is standard care, but we do insist that they're actually looked after in a high-risk pregnancy situation, thank goodness. They get their scans, but our wonderful Catherine rings them up every 2 weeks to make sure that things are going well, and if things aren't going well, then we're alerted to it. And then think about, well, what about the babies? What are we going to do with them? What's appropriate? And we spent a lot of time talking with our families about this, that basically they didn't want their kids over-medicalized, which would be so easy. They go through the standard NHS procedures, which are, the checks, the purple, the blue triangles are, the times when the health visitor or the time of immunization. They do have the carryover of mitochondrial DNA, because when you remove the pronuclei, you're almost bound to carry over a few mitochondria. So that's done at birth. At 18 months, they have a neurodevelopmental review, which is perfectly sensible because that's the time when most kids are walking and there's really good standards for that, and they're going to get something at 5 years. Here, my colleague Bobby McFarlane, who's a pediatric neurologist, is on speed dial for any medical problem. And in fact, of course, all children have medical problems. So he's pretty busy. Any coughs, they've sneezed, you can imagine, we better just check with Bobby. So he's very busy. And we have, funded by Wellcome, an 18-month follow-up of the children. So this is the assessment to see what is happening, and that's in progress. The other thing you have to realize is that this is carefully controlled. These patients, the pregnancies, have to have a child that is at risk of serious mitochondrial disease and not suitable for pre-implantation genetic testing. So these patients with heteroplasmy, and there's been at least 3 patients who've actually had PGT, 4 patients who've had PGT, who actually on testing all the embryos are too high to consider putting back, so they're going to go on the mitochondrial donation program. Each application has to be considered by the HFEA Statutory Appeal Approvals Committee. It's very important, this. So we might suggest it, but there is regulation. And the reason why there is regulation of this, and I've got to be a bit careful what I say, is the IVF world is the Wild West in some circumstances. There's an awful lot of practices where people are trying to improve so-called improved fertility with very little evidence, and people have suggested using this without any good evidence at the moment. So it's very carefully considered before we can approve it. So I thought I'd put this slide up. I don't know how many have seen this film. It's a really good Netflix film about the birth of IVF. It's really good. I really enjoyed it, and it just reminded me so much of our program. So much promise and so much disappointment, at least initially. I mean, it's very difficult, and I'll try and explain one of the reasons why we had such a problem to start with. So I explained, to do this, we actually, with the woman's eggs, we collect 3 IVF. They go through IVF 3 times. We need about, you want to try and get 20, 30 eggs. So that's a lot to go through. And you're really looking at egg number and egg quality. So associated with that, if you look at this curve, which is the number of follicles women have, and the quality of the oocytes, you see where there is a problem. We'd had a number of women who devoted 5 years of their lives to helping get mitochondrial donation through, and we wanted to help them. You can see where the majority of our first efforts were, and you can understand why it was so challenging to start with, because many of them were over 35. And now, I mean, we've actually moved the age limit from 40 to 35, because over 35, the chances of getting enough good quality oocytes for this to work is pretty low. I mean, I think this is a really important message that's come out of this, is discuss reproductive options with women with mitochondrial disease early. I think it's such an important message. And in fact, I mean, that slide which shows the curl off, we should all think about, or people should think about having their children earlier rather than later. I'm just going to use something theoretical here, because it is of public information that there have been births of children with following mitochondrial donation. That was announced by the HFEA back in 2003 as a public information request. They said it was somewhere between 1 and 5, and I can't really comment. I can say that obviously we're in 2025, sorry, it was 2023, we're now in 2025, and you can get your own inferences whether or not there's more now. I can't say too much, because quite simply, this is such a newsworthy story. And we are very, very concerned about the mothers who've got children with, who've gone through mitochondrial donation and have children, particularly now in this current social media background, where actually any keyboard warrior can write any sort of things to people. So we're being very cautious. The women are asked, or we suggest that people don't put things onto Facebook sites or other sites. So we're trying to be very cautious. I'm kind of priming you for when the papers do come out. But I just want to highlight one of the issues that could have come across. Let me just say that, okay? So, suppose one of the children going through mitochondrial donation had a medical problem. Let me just say a cardiac defect, structural cardiac defect. The difficulty here would be, well, is that due to the mitochondrial donation, or is it not? Because IVF is associated with increased risk, in particular of cardiovascular things. Mitochondrial disease itself is associated with increased pregnancy complications. Never mind the risk of embryo manipulation, because you can get the same sorts of problems in PGT, which now is not monitored at all, versus mitochondrial donation. And I suppose this is why I have a plea about translational medicine. What's done in science, and as I say, I'm going to put pictures up. My embryological colleagues have done fantastic science. But this is built on translational medicine. If there wasn't an outstanding clinician, my colleague Bobby McFarland, that was dealing with all the issues that are happening, if you don't have very good clinicians who are not only understanding the disease, identifying the solution, because if you don't understand that this is a problem in patients with mitochondrial DNA disease, how would you think of a solution? Patient care and selection, and expertise in managing medical problems. It's that unique skill set that this society in many ways represents, because it's so important that when we're thinking about the results of clinical studies and stuff like this, that this actually reflects something that is actually going to be of benefit to patients. I'd like to finish, of course, just as I showed the picture of the team that got through the things through Parliament. Of course, this is all teamwork. And I think that's, again, something great about academic medicine, is that we're able to work in teams. And it's my clinical colleagues and my embryological colleagues, Mary Herbert and Louise Hislop. And hopefully, I've been able to show you that this is something that's gone from conception to clinic. I hope that if I ever get invited back, I'll be able to tell you what the result is. I'm sure it'll come out within the next month or two, and at least you're pre-warned. Thank you very much indeed.